Good morning, students.
Morning, ma'am.
Good morning, ma'am.
Okay, so we'll start with the new chapter, that is protein digestion and absorption. Before going into metabolism, this chapter has to be covered how the proteins are digested and absorbed inside our gut. So all of you know that proteins are the macromolecules which are made up of amino acids linked by a peptide bond, isn't it? So the proteins have got different levels of protein structure. And when the protein has to be digested, first of all, what we do is we cook our food, isn't it? So there are protein-rich foods like milk, egg. These are non-vegetarian products like milk, egg, meat, and fish. And vegetable-rich proteins, you already know sources of protein, pulses, then beans, etc. So whenever you take this protein-rich food, it has to be first cooked, isn't it? So the cooking, what it will do, it will denature the protein. So what is denaturation? So during denaturation, the secondary, tertiary, and quaternary structure of the proteins are lost except the primary structure. So by denaturation, what will happen? All the proteins will be in their primary structure level. Okay, so these proteins are made up of amino acids. They are non-essential amino acids, then the essential amino acids and semi-essential amino acids. So non-essential amino acids are those amino acids which can be synthesized by our body, and we don't need to provide it in our diet. No, essential amino acid has to be provided in the diet, and semi-essential means they can be synthesized in our body, but the synthesis is not sufficient during growth of the child or during pregnancy. So you have to supply them in the diet. Clear? So roll number one, Aditya. Yeah. So just tell me what are the semi-essential amino acids?
Histidine and arginine are the same.
Yeah, histidine and arginine. Okay, they are known as semi-essential because they are required for the growth of the children and during pregnancy. Actually, they can be synthesized in our body, but they are not sufficient enough to sustain the growth and development. Clear? So when you take these different sources of proteins, these proteins are ultimately digested into amino acids. Now let us see how the protein digestion occurs. So just like in digestion of carbohydrate, the digestion starts in the mouth. Yes or no? Digestion of carbohydrate starts in the mouth. And what is present inside the mouth for digestion of carbohydrate? Roll number 2.
Ma'am, salivary amylase.
Salivary amylase, no? So for digestion of carbohydrate, you require a salivary amylase, and the digestion of carbohydrate starts in the mouth. Okay, but remember, digestion of protein, it cannot start in the mouth because mouth does not have the proteolytic enzymes. Okay, so the saliva does not have a proteolytic enzyme, so the digestion of the protein does not start in the mouth. Okay, now when this denatured protein, it reaches the stomach. The stomach, what does it contain? It contains the gastric juice. Yes or no? So what are the composition of gastric juice? Anyone? You have got, in the gastric juice, you have got the hydrochloric acid, then you have got the proteolytic enzyme, that is pepsin, and then you have got a mucus. So it is the mucus which lines the stomach, so it protects the cells which are present in the stomach against the hydrochloric acid that is secreted. If the mucus was not present, the hydrochloric acid will digest the cells which lines the stomach. Okay, so gastric juice composition is, it contains mainly hydrochloric acid, proteolytic enzymes like pepsin. Okay, then it has got renin in case of children, and also it has got mucus. Clear? Now when this denatured protein, it reaches the stomach, what will happen? This HCl, anyone who knows the cell which is responsible for the synthesis of HCl? What type of cells will synthesize HCl? Anup?
Peptic cells.
Parietal cells. Okay.
Parietal cells.
Yeah. Parietal cells secretes hydrochloric acid. Clear? And pepsin is secreted by pepsinogen because all the proteolytic enzymes are secreted in zymogen form. okay, inactive form, that they have to be converted into the active form. So pepsinogen is secreted by the what type of cells?
Parietal cells.
Parietal cells will secrete hydrochloric acid. Okay, the pepsinogen will be secreted by the chief
Peptic
cells.
cells.
Chief cells, isn't it? Chief cells of the stomach. Okay. Now there has to be a stimulus for secretion of hydrochloric acid in the gastric juice. Okay, what are the stimulus? You can note it down. The stimulus for secretion of hydrochloric acid in the gastric juice is acetylcholine, okay, histamine, and renin. Not renin, acetylcholine, okay, histamine, and gastrin. So gastrin is the hormone which is synthesized or secreted by the antrum of the pylorus. Okay, it is also secreted by the intestine. So what this gastrin does, it increases the stimulation of the parietal cells, or it increases the proliferation of the parietal cells, and it also helps the parietal cells to secrete hydrochloric acid. So remember, hydrochloric acid secretion is stimulated by 3 factors. Number 1 is gastrin. What is gastrin? It is a hormone which is produced by the pylorus. You know the pylorus, no? That is the end of the stomach. So the antrum of the pylorus, then it is also secreted by the intestine. So this gastrin, what it does, it increases the hydrochloric acid secretion by the parietal cell, and also it causes the proliferation of the parietal cell. Okay. Then HCl is also, secretion is also stimulated by acetylcholine and by histamine. Clear? Now, once the hydrochloric acid is secreted because of these 3 factors, this hydrochloric acid, what it will do, it will convert the pepsinogen, which is secreted by the chief cells of the stomach, okay, to pepsin. So the HCl, what it will do, it will convert the pepsinogen, which is secreted by the chief cells of the stomach, okay, chief cells of the stomach, to pepsin. How pepsinogen is converted to pepsin? By cleaving the 44 amino acids from the N-terminal end of the pepsinogen. Okay, so this hydrochloric acid, it cleaves the 44 amino acids from the N-terminal end of the pepsinogen, and then the pepsinogen is converted into pepsin. Okay, for this activity, you require that the pH should be maintained between 1 to 2. So because of the hydrochloric acid, the pH of the gastric juice is acidic, around 1 to 2, and around that pH, okay, the hydrochloric acid will convert the pepsinogen into pepsin, and once a small molecule of pepsin is produced, this pepsin will then convert the pepsinogen into pepsin. In the beginning, you require hydrochloric acid, but once the pepsin is produced, the small amount of pepsin will facilitate the conversion of pepsinogen to pepsin. Okay, remember the optimum pH for activity of pepsin is around 2. And what is pepsin? It is a proteolytic enzyme, and it is an endopeptidase. What is endopeptidase? It will break the peptide bond from inside the protein molecule. Clear? Now remember, this pepsin molecule, it acts on the peptide bond which is formed by the carboxyl group of aromatic amino acid and methionine. These things you have to remember. Okay, the proteolytic enzyme which is present in the gastric juice is pepsin. And from where do you get pepsin? You get pepsin from pepsinogen. How do we get pepsin from pepsinogen? With the action of hydrochloric acid, which is produced by the parietal cells. Okay, so you get pepsin from pepsinogen by cleaving 44 amino acids from the N-terminal end. And once this pepsin is produced, the pepsin is an endopeptidase, and it will act on the internal peptide bonds of the particular amino acid. So you have to remember what are the amino acids where the pepsin will act. These are aromatic amino acids like phenylalanine, tyrosine, tryptophan, and methionine. Methionine is not an aromatic amino acid, but phenylalanine, tyrosine, and tryptophan, these are aromatic amino acid. Okay, and methionine is sulfur-containing amino acid. So with the help of pepsin, proteins will be broken down into smaller and smaller fragments, which are known as proteoses and peptones. Clear? So this is the digestion which occurs inside the stomach. So remember, in the mouth, protein digestion does not occur because the saliva does not contain any proteolytic enzyme. Okay, so the digestion of the protein begins in the stomach with the help of hydrochloric acid. So what are the stimulus for secretion of hydrochloric acid? There are three: gastrin, then acetylcholine, and histamine. Okay, remember? So gastrin, what is gastrin? It is a hormone which is produced by the stomach and the intestine. And this gastrin, what it does, it causes the secretion of HCl as well as it causes the proliferation of the parietal cells. Clear? Now this pepsinogen is secreted by the chief cells of the stomach, and pepsinogen is a zymogen form. So conversion of pepsinogen to pepsin occurs with the help of hydrochloric acid. Okay, once a small amount of pepsin is formed, that pepsin will in turn convert the rest of the pepsinogen molecule into pepsin. Okay, what is the pH of the gastric juice? If somebody asks you, it should be around 1 to 2. Okay, and the optimal pH for the activity of pepsin is, pepsin acts on the acidic side, that is around 2. Clear? Now once you get the pepsin will act in the internal peptide bonds of the particular amino acid. It cannot go and break anywhere. Okay, it breaks the peptide bond which is formed by the carboxyl end of the phenylalanine, tyrosine, tryptophan, and methionine. Okay, by breaking these bonds, the protein will be converted into proteoses and peptones. Clear? Any confusion? Vivek?
No, ma'am.
Any confusion?
No, ma'am.
Oh, okay. ठीक है. So pepsinogen is secreted by which cells? हाँ विवेक बोलो.
Ma'am, no confusion, ma'am.
Okay, ठीक है. Pepsinogen is secreted by which cells of the stomach? कौन सा cells है?
Ma'am, chief cells.
Okay, and hydrochloric acid?
Ma'am, वो parietal और oxyntic cells.
ठीक है. तो how pepsinogen is converted into pepsin?
Oh, ma'am, by the action of HCl, pepsinogen is initially converted into pepsin, and then pepsin himself initiate the conversion of pepsinogen in pepsin.
Okay, ठीक है. Now this pepsin will act on the peptide bonds formed by which amino acid? It is there in the slide. It can.
वो phenylalanine, methionine, tyrosine, tryptophan.
Yeah, first of all, you remember the aromatic amino acids that are only 3, isn't it? Phenylalanine, tyrosine, and tryptophan, and then methionine. Okay? With the action of the pepsin, now proteins will be broken down into proteoses and peptones. Clear? Okay?
Yes, ma'am.
You can mute yourself. Then Chandani. Chandani, tell me what is the other proteolytic enzyme which is present in the gastric juice, which is present only in case of children, infants. Can you tell me the name of another?
Renin.
Renin. Okay, can you tell me what is the difference between RENNIN and RENIN? Yeah, both are same.
Renin.
Yeah, one is renin, another is also renin, isn't it? The pronunciation is same. So what is the difference? RENNIN and RENIN.
No, ma'am. One is, ma'am, RENNIN is a proteolytic enzyme that is present in infants.
Okay.
It's an enzyme, and the other renin is a hormone.
Okay, what is the function of the other renin?
That is present in the kidney.
Okay, what does it do?
Ma'am, vasoconstriction.
Okay, it converts angiotensinogen into angiotensin, isn't it? Angiotensinogen into angiotensin.
Yes, ma'am.
So angiotensin is the one which causes the vasoconstriction and increases the blood pressure, no? ठीक है? So remember, there is a difference. You can switch off or mute yourself.
Yes,
There is a difference between renin and renin. Okay, don't get confused. So renin is another proteolytic enzyme which is produced in infants. And renin, it is a hormone which is produced by the kidney. Okay. Now what this renin, what it does is that it converts the casein which is present in the protein, okay, milk protein, into calcium paracasein. Okay, converts the casein which is present in the milk protein to calcium paracasein or calcium paracaseinate, which helps in the curdling of the milk, okay, in infants. So this renin is absent in adults. Clear? So the proteolytic enzymes which is present in the gastric juice is pepsin in case of adults. In case of children, pepsin as well as renin is present. Clear? So renin is the proteolytic enzyme which helps in conversion of casein into paracaseinate, and thereby helps in curdling of the milk. On the other hand, renin, okay, it is a hormone actually, not enzyme, produced by the kidney. Okay, so what it does, it converts the conversion of the angiotensinogen into angiotensin. Clear? Now remember this renin, R-E-N-N-I-N, it is absent in adults. Now in the next step, what is the name of the acidic semi-digested product of the stomach known as, isn't it the semi-digested product which is present in the stomach, it is acidic, it is having an acidic pH, so it is known as chyme. So when it reaches the intestine, what will happen? Where the bile duct opens, in which part of the duodenum? The common bile duct, where does it open? Anatomy mein? Haan, Anup bolo? In the intestine, no? The common bile duct opens in which part of the duodenum?
Ma'am, the C-shaped duodenum.
Okay, the second part of the duodenum, isn't it? The common bile duct opens. So common bile duct is the duct which combination of the bile duct as well as pancreatic duct. ठीक है? Now, through the common bile duct, the bile acids. So in the bile, what is present? There is a sodium bicarbonate, isn't it? So the pH of the bile is alkaline. So when the bile is secreted into the duodenum, the pH of the duodenum becomes alkaline. Okay. Now the semi-digested product which is coming from the stomach, when it enters the intestine, the pH will be increased because of the sodium bicarbonate which is released in the intestine. Okay, released in the intestine through bile. Okay, the pH will be then increased up to 8. Why is it important to increase the pH? Because the proteolytic enzymes which also comes into the duodenum from the pancreas, the optimum pH is on the alkaline side. Okay. Now this pancreatic juice, what are the proteolytic enzymes which are present in the pancreatic juice? There are 4. Okay. All are secreted in the zymogen form. So the pancreatic enzyme contains important endopeptidases, namely they are synthesized as trypsinogen, okay, not as trypsin, trypsinogen, then chymotrypsinogen, then proelastase, then procarboxypeptidase A and B. Okay, procarboxypeptidase A and B. So these are the 4 hormones. Even collagenase is also produced by the pancreas. So these are the main important enzymes which is produced by the pancreas. That is trypsinogen, chymotrypsinogen, then elastase and proelastase and procarboxypeptidases A and B. So remember, all these are serine protease. Why they are known as serine protease? Because their active site, it contains the serine residues. Clear? Active site contains the serine residues. Okay, Jessica? So can you tell me what are the proteolytic enzymes that are produced by the pancreas? Name the proteolytic enzymes produced by the pancreas, Jessica. Jessica? Absent. Ryan?
Yes, ma'am.
Yeah, tell me the name of the proteolytic enzymes produced by the pancreas.
Trypsinogen, chymotrypsinogen, procarboxylase, proelastase.
Okay, okay. Procarboxypeptidases A and B, there are 2 types, remember, procarboxypeptidases A and B, then proelastase, then trypsinogen and chymotrypsinogen. Okay, these are the 4 proteolytic enzymes which are present in the pancreas and secreted by the pancreas. Now, trypsinogen is the inactive form of the trypsin. How it is converted into trypsin? Trypsinogen is converted into trypsin by the enterokinase. Okay, what are these enterokinase? These are enteropeptidases which are produced by the intestinal microvillus membrane. So enterokinase, which is the enzyme produced by the villus cell, microvillus cell, it helps in the conversion of trypsinogen into trypsin. So how it is converted? By removing 6 amino acids, okay, from the N-terminal end. So once trypsinogen is converted into trypsin with the help of enterokinase, okay, now trypsin will in turn help in the conversion of the chymotrypsinogen into chymotrypsin, then proelastase into elastase, then procarboxypeptidases A and B into carboxypeptidases A and B. Okay, so the stimulus for conversion of trypsinogen to trypsin is enterokinase. Okay, what is enterokinase? It is the enzyme produced by the villus cells of the intestine. Clear? Now once trypsin is formed, what it will do, it will help in the conversion of the rest of the proteolytic enzymes into their active forms, which are produced by the pancreas. Clear? Like conversion of chymotrypsinogen into chymotrypsin, okay, proelastase into elastase, and procarboxypeptidases A and B into carboxypeptidases A and B. So what happens in acute pancreatitis? There is premature activation of trypsinogen inside the pancreas itself, which will result in the autodigestion of the pancreatic cells, resulting in acute pancreatitis. Remember, acute pancreatitis is a life-threatening condition. Okay, so a person can die because of acute pancreatitis. So what happens there in acute pancreatitis? There is premature activation of trypsinogen to trypsin within the pancreas itself, which causes autodigestion of the pancreatic cells. Clear? Now, trypsin, it will act on the protein which is semi-digested in the stomach, and then remember it will act on the peptide bonds which are formed by the basic amino acid. I may ask any of the students, okay, the name the amino acids where these proteolytic enzymes will act. So trypsin, remember, it will act on the basic amino acid, that is arginine and lysine. Clear? So trypsin will act on arginine and lysine, and chymotrypsinogen. So remember this one. Pepsin, where it will act? It will act on the peptide bond formed by the carboxyl group of the aromatic amino acid as well as methionine. Trypsin, it will act on the basic amino acid, that is arginine and lysine. Chymotrypsin will again act on the aromatic amino acid, okay, aromatic amino acid as well as methionine. Elastase, it will act on the neutral amino acid, that is glycine, serine and alanine. Okay, then carboxypeptidase A stands for aromatic amino acid. Clear? And carboxypeptidase B stands for basic. Clear? Carboxypeptidase A will act on the aromatic amino acid. Carboxypeptidase B will act on the basic amino acid. Elastase will act on the neutral amino acid, that is glycine, serine and alanine. Clear? Chymotrypsin will act on the aromatic amino acid. Trypsin will act on the basic amino acid. So with the help of all this proteolytic enzyme, trypsin, chymotrypsin, then elastase, then carboxypeptidase A and B, the proteins or the proteoses or peptides will be broken down into smaller peptides, okay, tripeptide, dipeptide or individual amino acid. So this trypsin, chymotrypsin and elastase, remember they are endopeptidase, they will act on the internal peptide bonds. Carboxypeptidase A and B are the exopeptidase, they will act from the carboxyl end and will release the amino acid one by one from the carboxyl end. Okay, so with the help of these pancreatic enzymes, the protein will be ultimately broken down into individual amino acid, tripeptide and the dipeptide. Clear? So this, what type of enzyme is it? Carboxypeptidase, they are metalloenzyme requiring zinc. Okay, zinc is also present in carbonic anhydrase, isn't it? So carboxypeptidase is also a metalloenzyme requiring zinc. So in the duodenum, the digestion occurs because of the pancreatic juice. And remember, the secretion of the pancreatic juice, okay, it is stimulated by cholecystokinin, okay, which was also known as pancrealzymin. So cholecystokinin, what it does, it helps in the secretion of the pancreatic juice into the intestine, okay, cholecystokinin. And then there is another thing, secretin. Secretin will help in the secretion of the bicarbonate, sodium bicarbonate, into the intestinal cells, into the duodenum. So with the help of cholecystokinin and secretin, what will happen? The pancreatic juice will be released into the duodenum, as well as the sodium bicarbonate, which provides the alkaline pH for the activity of all these pancreatic enzymes. And ultimately, they will be digested into amino acids, tripeptides and dipeptides. Yeah. So till here, any confusion? Yeah. Okay, so protein digestion, the semi-digested product which comes from the stomach, it is acidic. Okay, so the pH will be increased because of the sodium bicarbonate which is present in the bile, and the release of the sodium bicarbonate into the intestine will be helped with the help of secretin. Then there is another hormone known as cholecystokinin. This cholecystokinin will help in the release of the pancreatic juice into the duodenum. Okay, so inside the duodenum, digestion of the protein will continue with the help of pancreatic enzymes, and there are four main type of pancreatic enzymes which are secreted in the zymogen form. Then this trypsinogen converted into trypsin with the help of enterokinase. Then the rest of the enzymes will be converted into their active form with the help of trypsin. Okay, and what will be the end product of digestion? You will get amino acid, tripeptide, and dipeptide. Clear? Then the digestion of the protein will continue in the intestine. Okay, in the intestine, and the intestinal juice is known as succus entericus. Remember, the intestinal juice, it is known as succus entericus, and this intestinal juice, it contains proteolytic enzymes. These are aminopeptidases. Okay, mainly the proline and glycine aminopeptidases, which releases the amino acid from the N-terminal end. And the succus entericus also contains dipeptidases and tripeptidases, okay, which will act on the dipeptide and the tripeptide to release the individual amino acids. Okay, so let us recapitulate the things, whatever was taught. So the gastric juice, what does it contain? It contains the pepsin. Pepsin is what type of enzyme? It is an endopeptidases. Okay, what is the who helps in the conversion of pepsinogen into pepsin? It is the HCl. Okay, the pepsin will act on the peptide bonds, internal peptide bonds, which are formed by phenylalanine, tyrosine, tryptophan, and methionine. What is the end product? Proteoses and peptones. Another proteolytic enzyme is renin, which is present in infants. Okay, the HCl. Okay, it will help in the activity of renin, and renin in turn will convert the casein into paracaseinate, which will help in the curdling of the milk. So for the activity of the pepsin and renin, you need the HCl because these enzymes will act on the acidic side. Pancreas, you have got trypsin, chymotrypsin, elastase, carboxypeptidases. Okay, and what is the end product? Proteoses, peptones, and peptides. In the intestinal juice, what are the proteolytic enzymes? Aminopeptidases, tripeptidases, and dipeptidases. Clear? So ultimately, you will get the end product of protein digestion will be dipeptides, and you may have some tripeptides and the free amino acids. Clear? Any confusion? Students? Vaishnav is there?
Yes, sir.
Yeah, bolo. From the
Yes, sir.
beginning till the end. Beginning till the end. Just tell me the enzymes which are present in the gastric juice, proteolytic enzymes.
There is gastric juice. There is
Yeah.
pepsinogen
Okay.
and renin.
And renin. How pepsinogen is converted into pepsin?
With the presence of HCl.
Okay. Then in the pancreatic juice, what does it contain?
Trypsinogen,
Okay.
chymotrypsinogen,
Okay.
and then procarboxy, something A and B.
Procarboxypeptidase because it acts on the peptide bond, no? Procarboxypeptidase,
Yes.
A and B. Then?
And B.
Then proelastase.
Proelastase.
Okay. How how trypsinogen is converted into trypsin?
By the enterokinase.
Okay. With the help of enterokinase, which is an enzyme which is produced within the intestinal cells. Clear? Now, how this chymotrypsinogen is converted into chymotrypsin?
With the help of trypsin.
Okay.
By the
With the help of trypsin. Okay. Similarly, the procarboxypeptidases and proelastase is converted into elastase and carboxypeptidase with the help of trypsin. Then what are the enzymes which are present in succus entericus?
Succus enterokinase, aminopeptidase.
Okay, aminopeptidase, then tripeptidase,
Dipeptidase.
dipeptidase. Okay, so these are the three proteolytic enzymes which are present in the intestinal juice. Okay, what is the pH of the gastric juice?
1 to 2.
2. So around that pH, the pepsin and renin will act. What is the pH of the this one around in the duodenum?
8, around 8.
Okay, ठीक है. So around that pH, these pancreatic enzymes will act. So the pH will be on the alkaline side in the inside the intestine. Okay, because of the sodium bicarbonate which is released through the bile. Clear? Any confusion? So ultimate end product of protein digestion is, remember, free amino acid. Then you have some molecules of dipeptide as well as tripeptide. Clear? You can mute. Now once you get this amino acid, dipeptide and tripeptide, it has to be absorbed. Yes or no? Like carbohydrates, no, starch, when it is broken down, you ultimately get maltose and you get glucose, isn't it? Now glucose, how it is absorbed inside the intestinal cell? Anup, you remember how glucose is absorbed inside the intestinal cell?
Active transportation.
What type of active transport was that?
Sodium.
Yes, sodium dependent active transport, which is secondary active transport, isn't it?
Yes, ma'am.
Yeah, so along with the sodium, the glucose is transported. Okay, similarly, you can mute yourself. Similarly, in case of amino acids are also absorbed. There are different mechanisms how these amino acids are absorbed. So one is by secondary active transport. The sodium concentration, you know that outside is more when compared to inside the cell. So sodium will move down the concentration gradient. Because of that, it will carry the amino acid along with it. Okay. It will carry the amino acid along with it. So this is known as secondary active transport because it uses the ionic gradient of the sodium. So amino acids are absorbed inside the intestinal cell with the help of secondary active transport, which uses the ionic gradient of the sodium. Okay, now once this sodium is inside the cell, the concentration will go on increasing, isn't it? So you have to remove the calcium from inside the cell to outside with the help of sodium potassium pump. Clear? So these amino acids can be absorbed with the help of sodium dependent amino acid transport, which is the secondary active transport. And remember, there are different transporters for different amino acid. You have got the acidic amino acid transporter, which is present in the intestine. Then there is a basic amino acid transporter, neutral amino acid transporter. There is a transporter for amino acid as well as for the glycine. Okay. Then there are transporters for beta amino acid. And remember, absorption of L amino acid is faster when compared to the D amino acid. So you have got different types of transporter for different amino acids. And with the help of this transporter, okay, and the sodium ion, this amino acids are transported with the help of secondary active transport, that is sodium dependent symport system. Clear? Then there is another way the amino acids can be transported inside the intestinal cells and also inside the liver cells, inside the kidney and inside the brain cells. There is something known as Mr. cycle. Okay, Mr. cycle is nothing but it is a gamma glutamyl cycle. Okay, gamma glutamyl cycle. So let us see what is this gamma glutamyl cycle. So this is an amino acid which is present in the intestinal lumen. Okay, this is the intestinal cells and this is inside the intestinal cell. So let us see how this amino acid which is present in the lumen, which is released after the digestion of the proteins, how it is absorbed. So what happens is that this glutathione, what is glutathione? It is a tripeptide, remember, which is made up of glutamate, cysteine and glycine. It is a favorite question of the examiners, okay? So they will ask you what is glutathione. You have to tell it is a tripeptide made up of 3 amino acids, that is glutamate, cysteine and glycine. Clear? The other name of glutathione is gamma glutamyl cysteine glycine. Clear? So this glutathione, it will react with amino acid and it will form a glutamyl amino acid. Okay, glutamyl amino acid, and the cysteine and the glycine part of the glutathione will be released as cysteine glycine. Okay, so when glutathione combines with amino acid, it forms glutamyl amino acid and cysteine glycine. Clear? Now this glutamyl amino acid, with the help of one molecule of ATP, okay, with the help of one molecule of ATP, The amino acid part will be released. Okay, the amino acid will be released inside the intestinal cell, and the glutamate will be left. Okay, this occurs with the help of the enzyme gamma glutamyl transpeptidase. Okay, gamma glutamyl transpeptidase. So you know that gamma glutamyl transpeptidase is a marker of what? Anyone? GGT. You remember it is a marker enzyme for which disease?
Chronic alcoholism.
Okay, good. Okay, so GGT is the marker enzyme for chronic alcoholism. Okay, so this gamma glutamyl transpeptidase, what it does, it converts the glutathione. Okay, glutathione. It breaks the glutathione into glutamyl amino acid and cysteine glycine. Okay, now once this glutamyl amino acid is formed, okay, the amino acid part of this glutamate will be released, and then again the glutamate will combine with glycine and cysteine to resynthesize glutathione. So this is known as gamma glutamyl cycle or Mr. cycle. Okay, so remember this gamma glutamyl cycle is helpful for the absorption of the neutral amino acids inside the intestinal cell as well as inside the brain cells and the kidney cells. Clear? So one important enzymes which is in gamma glutamyl cycle is gamma glutamyl transpeptidase. You can make out from the name. It breaks the peptide bond of the glutathione, and the glutathione will be converted into glutathione glutamyl amino acid as well as cysteine glycine. Later the amino acid will be released from glutamyl amino acid with the help of ATP, and then the glutamate will recombine with glycine and cysteine to form glutathione. Okay, remember for the transport of one amino acid inside the intestinal cell, you require 3 molecules of ATP. Clear? You require 3 molecules of ATP. So you can see from your book again the Mr. cycle or the gamma glutamyl cycle. Okay, so this is another way how the amino acids can be transported. Then the, larger peptides like dipeptides and all, they can enter inside the cell, or the tripeptides can enter in directly through the cells or in between the cells. So sometimes what happens when the food particles, dipeptide or tripeptide, the larger peptides, when it enters inside the cells, it can cause food allergy. For example, some of the people are allergic to nuts, isn't it? Peanut, then cashew nut. That is because this protein particles of this nut, the larger protein particles, it can enter directly in between the cells, in between the intestinal cells, and then it is recognized as an antibody which generates an immune reaction, okay, leading to allergic reaction or the food allergy. So dipeptides and tripeptides can enter directly through the mucosal cells or in between the cells. Once it is inside the cell, it will be broken down into amino acid, and then from the intestine it will go to the liver, isn't it, through the portal system. Sometimes some of the dipeptide and tripeptide are immunogenic, then it can cause antibody reaction leading to food allergy. So there are 3 ways in which the amino acids and dipeptides can be transported. Amino acid by secondary active transport and by Mr. cycle, and dipeptides directly through the intestinal cells. Okay, now let us see some of the clinical conditions related to protein digestion and absorption. So this Hartnupp's disease, they used to ask some of the important things only I will mention. Hartnupp's disease. Okay, so Hartnupp's disease occurs because there is a defect in the transport of the tryptophan and the neutral amino acid. Okay, please note it down. Hartnupp's disease, what is the defect? There is a defect in the transport of neutral amino acid and tryptophan. Clear? Now you know there is one vitamin which can be synthesized from tryptophan, that is niacin, isn't it? 60 milligram of tryptophan gives to 1 milligram of niacin. So if there is Hartnupp's disease, what will happen? The tryptophan cannot be converted into niacin, and niacin deficiency causes pellagra, isn't it? What are the signs and symptoms of pellagra? Chandani, bolo. Signs and symptoms of pellagra.
Ma'am, I don't know.
Okay, okay. So pellagra, remember there are 4 D, capital letter D. Okay, 1 D stands for dementia, another D stands for dermatitis, okay, the 3rd D diarrhea, and the 4th D death. So there are 4 Ds in pellagra. It is very easy to remember. Diarrhea, dementia, dermatitis, and death. Okay. So in Hartnupp's disease, what happens? There is a transport defect. There is a defect in the transporter for the neutral amino acid as well as tryptophan. And remember, you can synthesize niacin from tryptophan. So in Hartnupp's disease, there is tryptophan deficiency or the niacin deficiency leading to pellagra. Clear? Then protein losing enteropathy, it can occur during bacterial infection or the viral infection. So what happens? Whatever protein you take, it cannot be digested and absorbed. As a result, excessive amount of the protein will be lost through the gastrointestinal tract. Clear? Then there are conditions which can affect the digestion and absorption of protein. I have already told one is protein losing enteropathy, which can occur in the viral infection like HIV or in the bacterial infections. Okay, and sometimes in case of partial gastrectomy, then pancreatitis, carcinoma of the pancreas, and cystic fibrosis, these conditions may affect the digestion and absorption of proteins. Clear? So these are some of the clinical application of digestion and absorption of protein. So I'll stop here. Okay, so whatever you have learned, you please read it again. So remember, digestion of protein does not start in mouth. It occurs in the stomach, it occurs in the duodenum and the rest part of the intestine with the help of the gastric juice, pancreatic juice, and the succus entericus. There are different proteolytic enzymes which are present in this different juice, and they act on different kinds of amino acid which you have to again revise. Okay, and they are converted into their active form by different stimulus. Okay, once they are digested into amino acids and dipeptides, they are absorbed with the help of secondary active transport. Okay, and with the help of Mr. cycle. And there are so many clinical applications of this in relation to digestion and absorption of protein. Mainly, you remember this Hartnupp's disease, then protein losing enteropathy. Then what are the conditions which can affect the digestion and absorption of protein? You have to remember partial gastrectomy, pancreatitis, cystic fibrosis. Clear? Clear? So that's all for today. So in the next class, we will start the protein metabolism. So once it is absorbed and enters inside the cell, different kinds of cell, whether it is inside the brain cell or whether liver cell or the kidney cells, these proteins will be broken down into amino acids, and most of the amino acids will be reutilized to form the protein. Some of them will be broken down into ammonia. Ammonia will be later converted into urea. So that is all brief about metabolism. Okay, so the proper metabolism will start in the next class. Okay, thank you so much. You revise again whatever has been taught in the class. Okay, thank you, students.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.